All right, welcome back, everybody. So what we're going to do now is head into the next section, right? Even though the exam 2 is today, we need to get started on the material for exam 3, which is going to be in a couple of weeks' time. I think July 16th or thereabouts. So, so this lecture today is a fairly straightforward one, so we may end up finishing this early, which wouldn't be a terrible thing. And if we do, then we'll just start lab earlier too, right? So that we kind of make the most of that. All right. Yeah, sorry, fine with Zoom stuff. All right, so essentially what we're going to do now is start kind of talking a little bit about kind of slightly bigger picture stuff. This lecture is mostly about kind of putting into context what's going on at both sort of like an evolutionary level, I guess, in terms of genome size. Hang on. I got a feeling that that's the garage about my car. And then really talking a lot more about kind of chromosome dynamics in terms of what's going on in the individual chromosomes, what those chromosomes look like in the nucleus, which isn't really kind of what you think it looks like, which is kind of cool. So when we want to visualize chromosomes, we always kind of get them in a condensed state, right? So this is going to be typically, well, this is most likely mitosis, right? So if you look real careful at the X, for example, you can see that there's two sister chromatids kind of together. And also if you look kind of carefully in the others, right? So these are condensed chromosomes. You wouldn't be able to distinguish them otherwise, right? They would just be a kind of translucent mass, right, of DNA. It would be very hard to visualize. And these have also been labeled with fluorophores, right? So fluorophores are proteins or chemicals which fluoresce, right? And they're very cool. It's kind of actually like chlorophyll, or even, you know, fluorescent lights or the phosphor in the LED lights that you might have in your home. And so essentially what happens is an electron gets excited by an incoming photon of a particular wavelength. And when it relaxes back down again, it re-emits that energy at a different wavelength, right? So these are, I'm not entirely sure what fluorophore is being used or what they're labeling, but there's one way of visualizing what's going on at a cellular level, something that I use quite a lot. We'll have a look at some pictures later, which I've been just finding tooling around the internet. But for now, this is just how we do it. And so essentially what we do is we order the chromosomes based on size, right? So chromosome 1 is always the largest, and then the smallest one isn't necessarily the smallest autosome, but the smallest autosome has the largest number, essentially, right? It's kind of how they put in order. So this would be a kind of a karyotype from a, or yeah, kind of karyotype is what it's called, from a human, because we've got 23 chromosomes and a sex chromosome, right? Or 23 pairs of chromosomes, 22 autosomes, pairs of autosomes, and 1 pair of sex chromosomes. And so this is from a male, obviously. Okay, so one thing that's kind of cool to talk about is eukaryotic genome size, right? So we're about 3 billion base pairs, right? So this is our haploid genome. So our actual total genome is twice that, right? It's about 6 billion base pairs. And it's all grouped in like different things. I don't actually even know what half of these things are, to be honest, because I'm not much of a taxonomist. But you can see there's a huge range in genome size, and we're actually fairly kind of well in the middle of that. Right? And so you have plants actually are often really large, right? So angiosperms and gymnosperms can have, easily, twice, 3 times the size of our genome. I think the largest one that I know of is wheat. And wheat has a genome of about 100 billion base pairs, or 110, something like that. So a whole order of magnitude larger than our own genome. And yet you don't see, wheat plants kind of ruling the world and building airplanes and stuff. And so this is really kind of a cool pictorial representation of what's called the C-value paradox or C-value enigma, right? The size of the genome has absolutely nothing to do with the complexity of the organism, right? It has a lot to do with other stuff, but not kind of where different organisms are at in terms of evolution. And we're down here in mammals. So mammals have a pretty small range. Bony fishes, really large for some reason. I don't know why. And so just a few units, right? So we're talking about kilobases, it's 1,000 nucleotides, megabase is 1,000,000, gigabase is 1,000,000,000. And you can see here we've got Takifugu, right? So this is pufferfish, made famous by a Simpsons episode, if anyone's ever seen that, where Homer trains to become a pufferfish chef, sushi chef. Anyway, most people probably haven't seen that. And whatever that is, which looks like a skink without legs. Maybe it is a skink without legs, I don't know. But you can see there's a huge range in genome size between these two, right? From 400 megabases to 90,000 megabases. But when you look at them, there's not really any notable difference in complexity of the organism, right? It doesn't really stand out. And so that's what's called the C-value enigma, right? Basically what it means is that there's no correlation between genome size and organismal complexity. There's not even a relationship between gene number and organismal complexity. So for example, C. elegans, my favorite model organism, which has 939 cells. And it has around 20,000 genes. Homo sapiens, if I can spell that right, I don't know, millions of cells, I use a zoom, etc., around 20,000 genes, give or take. And so there's no correlation between how many genes you have and organismal complexity. It's a lot more complex than that. And in fact, C. elegans has far more different types of genes than we do. So it has, hundreds, I think around 300 what are called nuclear hormone receptor genes, right? And they're basically involved in cell-to-cell signaling, right? We have 7 or 8. So the particular ecological niche that C. elegans has expanded to fill has selected for multiple rounds of duplication of those particular genes because that's what is has the highest impact on its fitness. Whereas humans, doesn't matter so much, right? So the only correlation that I've ever been able to figure out, bless you, is organismal oops, complexity, for want of a better word, and chromosome number. So typically higher mammals have more chromosomes than lower animals. And that really just reflects evolutionary time. So chromosomes merge and fragment over evolutionary time. And so, you know, if enough of that time and selection has gone on, then you'll end up with different species with different chromosome numbers. And that's pretty much it, to be honest. There's no other correlation that I can figure out. And so the big question is, okay, well, so if we have, oh, and also only, I don't know, 3 to 5% at most encodes protein coding genes. So the things that do stuff in terms of proteins, those that DNA sequence covers 3 to 5% of our genome. So there's an awful lot of other stuff there, right? A hell of a lot of stuff. And this has always been considered junk, which is a kind of a pejorative term in terms of DNA. But there's a lot of just genetic baggage that we carry around with us, right? And so an awful lot of that genetic baggage is repetitive sequences, right? So some of those repetitive sequences serve a purpose, as in the telomeres, right? They're full of repetitive sequences. Others are just what we kind of accumulate over time, right? And so a lot of these spots are basically short nucleotide sequences, so 2 to 3 bases quite often, that are repeated over and over and over and over and over again, typically as a result of errors during DNA replication. Now, sometimes these expansions can actually cause disease if they are in a protein coding region in a gene. So ALS, or Lou Gehrig's disease, is actually caused by a repeat, it's called a polyglutamine repeat. So it's a glutamine codon that's repeated over and over and over again. And once you get to enough repeats, then you start essentially having neurodegeneration and Lou Gehrig's disease. But most of these repeats are in just swathes of our genome that don't encode genes, right? Of which there's an awful lot. And these are generally called, in a kind of group term, something called satellite DNA. Right, so it's kind of in many ways the DNA that's at the periphery in terms of function. Right, some of this stuff actually does have function. We just don't know what it is yet. Others is just kind of like ancestral relics that we just carry around with us because there's no pressure to get rid of them. Right, they're neutral in evolutionary terms. And so obviously this is one explanation for that C-value paradox, right? Organisms that have smaller genomes may simply have less of this repetitive satellite DNA than organisms that have more, right? And so one thing that you will actually see is that I think is pretty cool is that organisms that are under pressure to reproduce quickly and in adverse environments like parasites, for example, they actually tend to have fairly small genomes, right? Because obviously the larger your genome, the longer it takes for you to copy your DNA and the longer it takes for you to be able to reproduce. So that's kind of another way of thinking about it too, which is kind of neat. Put it into context. And so when we look at these repeats, typically there's single copy sequences, and those include genes, as well as whether protein coding or RNA coding, or non-coding as they're called, and other just bits of DNA sequence could be anywhere from like a third to 3/4 of the genome. And then you have repeat sequences, right, depending on, kind of classified depending on how many times they have been repeated, right? And you'll see that, up to almost half of an organism's genome could consist of simply highly repetitive sequences, stuff that's just repeated over and over and over and over thousands of times. And this varies from organism to organism. And when we talk about transposons, which I think will be Thursday, we'll have a see, and actually one of the causes of this repetitive sequence is transposons jumping around in our genome, which is kind of neat. DNA pirates, basically. And so another thing that is kind of curious, and this in some way I presume represents the evolutionary history of these genomes, is that genomes are not equal in terms of chromosomes are not equal in terms of gene density. So you can have chromosomes which have, I don't know, as low as like single-digit genes per megabase, per million bases. And, so chromosome 4, 5, 18, so on, have less than 10 genes per million base pairs, right? So that's like 1 million base pairs, and you'll find, a few genes in there. Other chromosomes are the opposite. Which one's the most densely packed? 17, number 19, right? It has 27 genes per million base pairs, right? So it has almost the same number of genes on the chromosome as chromosome 3, which is 3 times larger. And actually the Y chromosome is the one with the lowest density of genes of all. And part of the reason for that is because they are genes that have really only a role in male development. So there's a huge variation. However, whatever the case, DNA is a really large molecule. What is it if you get all of the DNA from every single cell in your body and you stretch it out, it can reach the moon and back or something like that? Some kind of crazy amount of DNA. And nuclei are tiny, right? In the range of, 5 to 10 microns. So that's millionths of a meter, whatever that is in inches, small, right? I think that's 40 ten thousandths of an inch. So anyway, really small. And so the issue is fitting all of that DNA into that space, but being able to access it as well, right? So it's kind of like an kind of balancing act. And one thing that really, really want to get across is that genomes are dynamic, right? DNA is not just sitting there stationary in one single state. It is constantly changing from being open and active to being closed and inactive on a cell by cell and tissue by tissue and chromosome by chromosome basis, right? There is a constant dynamism, flow between different states of different chromosomes, right? And obviously this varies over your body, it varies over your lifespan, it varies over your development as well. And so there's this issue of, how do we fit all this DNA in there, and how do we make it available for transcription you know, if we're a cell in stationary phase, or DNA replication if we're in the cell, you know, in cell division. And so part of the way in that's dealt with is simply by coiling it more tightly, right? So this is called supercoiling. Again, an issue that we encounter in DNA replication ahead of the replication fork, right? And so as with that, and DNA gyrase is a type of topoisomerase, you can actually use topoisomerase to induce more supercoiling, right? So you can kind of pack the DNA in even more tightly. But the main way in which we manage this is through the formation of chromatin, right? And chromatin is protein and DNA. So it's a complex of the two. And the main chromatin protein is the histone or histone proteins. And histone proteins are highly conserved and they are extremely important. So actually kind of flipping it on its head, the reason why genes are highly conserved, and so this is kind of always think of evolution as being something that you stand and you look back at, right? You can't look forward in terms of evolution. You can only look to see where you have come from, right? And if a gene is highly conserved, that means that it doesn't differ very much between different species or taxa. So it doesn't differ much between, you know, bony fish versus cartilaginous fish, or, you know, fish and mammals and whatever, right? And so the more conserved that gene is, basically the more critical it is for the function of that organism. And so what that's telling you is that typically any mutations that occur in that gene are bad. And so natural selection has gotten rid of most of the mutations that naturally occur in the histone genes, so that when we look at them across taxa, across species, they look very, very similar, right? And so that just goes to show how important these proteins are. And essentially, the DNA is wrapped around these histones to form a nucleosome, right? Which is the bead on the string. It's the repeating unit of chromatin. And so this is the whole kind of beads on the string kind of model. And it's actually a real term. It's not just one that we make up. And each one of these is a nucleosome. And so essentially, when we talk about a nucleosome, we're talking about the DNA that's wrapped around the histone proteins. And we're talking about typically from the beginning of one nucleosome to the beginning of the next one. So I'm just going to put some terms in here. Right. And so that, you know, a nucleosome basically equals around 200 base pair of DNA plus 9 histone proteins. 4 pairs in the core particle. Plus 1 histone 1 outside. Now, if you, and essentially what this is showing is if you kind of chew up the DNA, typically those enzymes will chew up the DNA that's available, right? So that's going to be what's between the nucleosomes. Now, if you kind of try even harder, what you'll end up with is just the DNA wrapped around those 8 histones, right? So the core particle equals, again, it varies from species to species, but about 140 base pair of DNA plus the 8 histone proteins. And so the bit of DNA between, which is around 50 base pairs or so, that's what actually allows the whole thing to twist and flex and move and wrap around itself more tightly. And histone 1 acts to hold everything together and fold the chromatin. And so when you look at that in terms of this is kind of what it looks like when you blow it up even further. So we have 4 pairs, H2A, H2B, H3, and H4. A total of 8. And we have about 145 base pairs wrapped around those 8 histone proteins. And then between each core particle, we have about 50 base pairs, give or take, and which is often called linker DNA because it links the 2 core particles together. And our histone H1 protein binds to both that and the core particle. So whenever you read about epigenetics and histone modifications, they'll always preface which histone they're talking about by these numbers. And so there's actually like a histone code. And that's basically about which histone is modified, where, under what context. So you can change the properties of histones by modifying them. And we'll get to that when we look at kind of big picture gene expression stuff, which will be, I think, from memory in the last section, which is a really big part of how we control gene expression in eukaryotes. But we're not going to go into that just yet. And so you typically don't find much DNA in the nucleus in this form. You'll never find it bare, right? As soon as DNA is replicated, it is wrapped around these histone proteins and packaged back into chromatin, right? So you never find bare DNA, very, very rarely, and even if you do, only little bits of it. You typically find DNA in this kind of more compacted state. Sometimes it could be spread out, but typically you'll find it more compacted. And so basically what you can see is that those beads on the string have been wrapped around and coiled around each other. And that creates a chromatin fiber, essentially. And you can kind of see that progression here, right? So if our DNA double helix is about 2 nanometers, you know, chromatin fibers typically are around 30 to 300 nanometers, by and large. And so this packaging is the typical state that you'll find DNA in a non-dividing cell. However, the only time, let's write this down for posterity. You see the classic X-shaped chromosomes is in cell division. Right. You'll never see cells that chromosomes that are this tightly compacted and packaged during normal cell function. Right. This is very much a specific state limited to M phase, the division of nuclear DNA. So another few words to talk about, which we're going to use a bunch coming up, is euchromatin versus heterochromatin. So euchromatin is basically more loosely packed or packaged, more open to transcription. So basically it's transcriptionally active. Heterochromatin, on the other hand, is the opposite. Yeah, if I could spell properly. It's tightly packaged, very little to no transcription occurs. And this varies chromosome by chromosome, arm by arm, even region by region on the chromosome. And there's a constant dynamic between the two states for most of each chromosome. Right, so areas of the chromosome might be opened up, transcription active, the needs of the cell change, they can then be shut down again, back into heterochromatin. Bless you. And also what's kind of cool to kind of learn is that different chromosomes have their own spot in the nucleus, right? So they're actually chromosome territories in each nucleus of a non-dividing cell, right? So these different colors each represents a different chromosome. And those chromosomes are actually tethered on the inside of the nuclear membrane. So if you think of the nucleus as a ball, right, on the inside surface of that ball are anchor points, and those chromosomes are actually stuck to that. Allergic genetics or allergic to genetics? Probably better not answer that. I just blame all my cats and dogs. Right, and so you can actually see, and you can visualize. I keep trying to find good videos of this, but it's really tough to kind of ferret them out. But you can actually see different regions of each chromosome become active and then become inactive over time. to visualize that. I was trying to find some good pictures. This is actually of visualizing chromosome. This isn't quite the same, but this just shows you how you can actually visualize chromosomes. And so in a normal, this is about Patau syndrome. I think that's trisomy chromosome 13. But you can see that chromosome 13 has been labeled, right? That's where it kind of hangs out in the nucleus. In a patient with Patau syndrome, you actually see the three chromosomes here that are labeled. So it's not really quite the same, but it's still a cool picture. So you can actually visualize where these chromosomes are. And these are actually visualized using CRISPR live fish. Huh? I love these fancy names. So FISH is fluorescent in situ hybridization. It basically means that a fluorescent probe is sticking to something and glowing, which is a kind of cool technique. Anyway, I'll keep looking around for those pictures because I've seen them every once in a while. It's just hard to find them. And so the last thing, and this is actually a fairly short lecture today, and I'm going to call it quits in a little bit, is that the heterochromatin or chromatin state of DNA also varies by where on the chromosome you're looking at. So in the center and the tips, you'll typically find heterochromatin, right? These are transcriptionally inactive regions. They actually don't contain very many genes anyway. Right? If you remember back to when we were looking at map distance, right? Map distance doesn't always equal physical distance. Oh, there's a cat. That is a funny looking cat. Very cute. I think I'll be constantly blowing cat fur out of my nostrils if I had a cat like that. Mine are all short hairs. I just have a lot of them, hence cat fur everywhere. So the centromere and the central regions of the chromosome tend to have a lot of satellite DNA, have relatively few genes, right? Which is why map distance doesn't equal physical distance for the center of the chromosome. And tend to be pretty much transcriptionally repressed, right? The genes that are present there don't tend to be very transcriptionally active. And the telomeres don't have any genes whatsoever. They just repeat DNA. Actually useful because that's what's used to extend the telomeres in like germ cells and the like. But there's no genes there. Whereas in the arms, right, the bits kind of in between those two, you'll find a lot of genes. Generally speaking, it's going to be euchromatin and transcriptionally active, but that obviously depends on chromosome by chromosome. Right. So the arms will only be compacted down during M phase, which is essentially when it looks like this. Normally, it'll just be this huge long string, right, kind of clumped up in a particular region of the nucleus, right? It wouldn't look like that kind of classic egg shape. So yeah, that's pretty much all I have for today's lecture. It's a pretty short one. Normally I would continue on into another PowerPoint, but I think given that you have an exam today and tomorrow, you'd probably rather spend the time studying instead or doing other stuff. So, and also I need to pick my car up from the body shop too, so at some point before my kids' swim meet. So let's give it a half hour. If nobody has any objections, meet at 1:20 for lab. And Galen, that's kind of what I was talking about earlier. I had a feeling that we'd finish early because this is a fairly short PowerPoint. And it shouldn't be too long for lab. Should be like about an hour, give or take, from memory. So that should let us kind of get out and get on with other stuff a little earlier than normal. Then we'll pick up the usual schedule on Thursday again. Cool. All right, so see you all in half hour. Enjoy your lunch. And I'll see you in a bit. Pardon me.